Okay, hello, this is Bryan Roth from University of North Carolina, Chapel Hill, and what I thought I would do today is discuss DREAD technology as a general form of what's called chemogenetics. And what I'm going to show you today is how engineered G-protein coupled receptors can be expressed in distinct regions of the brain, uh, how they can be activated by a inert small molecule, and then how this leads to alterations in the firing of neurons and ultimately change in behavior in experimental animals and ultimately perhaps in humans. So the particular platform that we use relies on engineered G-protein coupled receptors. And as you may or may not know, G-protein coupled receptors represent the sector of the genome that is devoted to signal transduction. And of this, it's the largest in the human genome. There are perhaps close to 1,000 G-protein coupled receptors in the human genome, and they comprise about 4% of the proteins encoded by your genome. So a large hunk of your DNA is devoted to encoding G-protein coupled receptors. And of the G-protein coupled receptors in the human genome, most of them have their highest level of expression in the brain. So this is a heat map from a paper that was published by Jean Regard and Sean Coughlin in Cell some years ago, where they measured the expression of all the non-olfactory G-protein coupled receptors in the genome in various mouse tissues, and in particular in mouse brain. And as you can see, more than half of them had their highest level of expression in the brain. And there were striking regional brain distribution for these. And if you look at the link below, my website has basically all the raw data as well as individual heat maps for individual G-protein coupled receptors. So they're widely used in the brain for regulating the activity of neurons and other tissues, other proteins. Now, one of the things that's very interesting about G-protein coupled receptors, and something that my lab has been interested in for a number of years, along with many other labs around the world, is that they couple to multiple signal transduction pathways. And I'm sure most of you know that following agonist binding, there is dissociation of the heterotrimeric G protein, leading to the activation of various downstream effectors. In this case, we have a GPCR that couples to phospholipase C beta via G alpha Q. This leads to the hydrolysis of a particular membrane-bound phospholipid PIP2, the release of inositol trisphosphate IP3, and then the mobilization of intracellular calcium. And this particular pathway is called canonical G-protein coupled receptor signaling, and this is the pathway that gives GPCRs their name, since they're coupled to G proteins. Now, one of the things that was discovered now about 15 years ago by investigators in Bob Lefkowitz's lab was that after canonical GPCR signaling, there's phosphorylation of the receptor by G-protein receptor kinases. This leads to the binding of proteins called arrestins, which serve to arrest signaling through GPCRs through canonical GPCR signaling, but then serve as scaffolds for various downstream effectors. And what I've shown here is engagement of the RAF-MEK-ERK pathway, which leads to a different type of signaling. So generally, canonical G-protein coupled receptor signaling occurs very quickly, whereas this non-canonical signaling occurs relatively slowly. And the canonical signaling is basically what we've hijacked for our chemogenetic tools that I'm going to describe here shortly. Now, one of the other major advances that has occurred over the last few years and my lab has had some role in this, has been identifying various motifs within G protein coupled receptors that are responsible for specifying either arrestin or G protein signaling. And what I show here is simply a three-dimensional structure of a particular serotonin receptor that was recently solved by Daniel Wacker, who was a graduate student in Ray Stevens' lab, now a postdoc in my lab. And that paper, as well as a recent paper that was published in Nature by my lab and Ray Stevens' lab about 3 weeks ago, have begun to identify the structural motifs that are responsible for G protein signaling versus arrestin signaling. And in the future, I think we'll be able to design drugs that particularly activate those various pathways, and these will likely be useful for as therapeutic agents. As well, the emerging data regarding the structural motifs responsible for G protein signaling will allow us to create new chemogenetic tools that specify these various downstream pathways. So that's the future. So what I'd like to do with that as a background for G protein coupled receptors and G protein coupled receptor signaling is tell you initially a little story about a drug that my lab has been interested in studying for many years, and how this particular discovery led to the chemogenetic platform that we call DREADS, that I'm going to be discussing for most of my talk. And the story begins with now probably in the year 2000, when I was still practicing psychiatry. A student at Case Western Reserve University, where I was at the time, came to me who had smoked this plant here, Salvia divinorum, and he had had a very intense hallucinogenic experience. And he came to me because I was a psychiatrist, and he was concerned that perhaps he had had some irreversible damage to his brain, or that it might be inducing some psychosis in him. And I examined him and found, that he was fine, and just cautioned him not to take the drug again. But I was interested in what the drug was. And he told me it was this plant, and so I looked up some information about the plant, and it turned out that the active ingredient of this plant is this compound shown up here in the upper right, salvinorin A, and it's a lipid molecule, structurally called a diterpene, and it turns out that this is the most potent naturally occurring hallucinogen. So I was very interested in this, and since then, Salvia divinorum has become sort of a common drug of abuse in many places within the U.S., Canada, and elsewhere. It's not actually regulated by the law. And this is a photograph that was taken by a friend of mine, Roger Sunohara, where Salvia divinorum leaves were available from basically a convenience store in Canada. So it's fairly widely available. And after I began studying it, we realized that there were three basic questions that we'd like to know the answers to, and in each case, each of these questions required the development of completely new technology. And as well, each of them had increasing levels of complexity. So the first question was, what were the molecular targets or target responsible for the actions of salvinorin A? The second was, once we discovered the target, how is it that salvinorin A engages this target? And finally, how are the effects of this compound mediated on human consciousness? And I'm going to briefly discuss the first two technologies and then spend the rest of the time on the third technology, which is this chemogenetic platform that we've developed that has become broadly useful. So the first question we had was, what are the molecular targets? And to identify the molecular target, we developed what we call a parallel molecular target profiling platform. And basically, the way it works is this: it's a platform that was developed through a contract that I have with the NIH to develop small molecule-based screening programs. It's the NIMH Psychoactive Drug Screening Program. And basically what we have is we've developed a platform where we have large numbers of molecular targets, which are shown here. You can see G protein coupled receptors in various colors. And currently we have essentially every G protein coupled receptor in the genome available for screening. So essentially the world's largest collection of molecular targets. And basically what we do is, unlike what's done in the pharmaceutical industry, where a single molecular target might be used to screen 1 million compounds to find a single drug at a single target, what we do is we screen large numbers of targets in a parallel fashion using now robotics-based technologies and relatively small libraries of compounds. So it's basically the inverse of how things have been done in pharma. And the technology has been used and described in a large number of papers from my lab in collaboration with others, and you can see them listed there. So many recent papers in Science, Nature, Cell, and so on. So the platform works quite well. So we had basically an early version of this platform in the early 2000s. At that time, perhaps 100 molecular targets available for screening. And what we did simply was we just screened salvinorin A, so the purified plant compound, against a large number of molecular targets. And in comparison, we screened LSD. So LSD is a known hallucinogenic agent. And what we found, to our surprise, was that salvinorin A interacted with only one receptor in the genome. This is the kappa opiate receptor. Whereas you can see LSD has this very complex pharmacology. And so it was clear from that study, and then really hundreds of studies that have occurred since, that salvinorin A is a very potent and selective kappa receptor agonist. And these findings have been widely replicated since that time. Now, the thing that's sort of unusual about salvinorin A is it interacts with a single molecular target. And my lab then became very interested in discovering how it is that salvinorin A engages this target. And this was a considerably more difficult and continues to be a considerably more difficult question to address. And the long and short of it is, after about a 5-year collaboration with a number of talented individuals in my lab and in Ray Stevens' lab, we were able to collectively obtain X-ray crystal structure of the kappa opiate receptor. And this was published in Nature a little over probably 1.5 years ago. And in that issue of Nature, actually the structures of all 4 opiate receptors were solved. And unfortunately, this was an inactive state of the receptor with a kappa antagonist, and we're not yet at the stage where we can describe with any atomic level certainty how it is salvinorin A engages the target. But this data certainly gave us some very strong clues as to how this is, and this is an active area investigation now going forward for my lab. Well, the larger question, though, is, and this is the case really for all psychoactive drugs, as well as various drugs that are used for treating CNS disorders. How are these effects mediated in the brain? And for salvinorin A, the question was, how are the effects, these profound effects on human consciousness, encoded? And for this, we had to basically devise a completely new platform. And so the idea that we had was, what if we could make an engineered G protein coupled receptor, and we could create it in such a way that it could be activated by a ligand, which is pharmacologically inert. And then the idea was that we could, once we had created this G protein coupled receptor, perhaps through viral or genetic technologies, we could insert it into the cells that we thought might be responsible for a particular behavior. And then when we activate those, either activate or inhibit those cells with the G protein coupled receptor, we might recapitulate the behavior. So this was the idea. Now this is an idea, sort of a general idea that's been around for a long period of time. And so the idea has been recently resurrected with the BRAIN initiative. But really the genesis of this idea goes back to some very important papers that were published by Francis Crick in the 80s and 90s. And I just sort of highlight one of these. This was his last paper. So Francis Crick, of course, was responsible in part for identifying the structure of DNA in the 1950s. So one of the things that he said that we needed to do was to identify all the different types of neurons that exist in the brain. And then once we've identified them, we need to be able to turn them off or turn them on in a freely behaving animal in some sort of way. And of course, optogenetics has arisen as a very powerful way of doing this. Unfortunately, optogenetics is not a particularly useful platform for modulating G protein coupled receptor signaling or recapitulating the actions of drugs at GPCRs, really for a number of reasons that I'm not going to go into here. But we thought it might be useful to create, instead of optogenetic tools, what we call chemogenetic tools, which recapitulate the activation of G protein coupled receptors. And this is an idea that again has been around for a long time. And in fact, as early as 1991, Kathy Strader, when she was at Merck, was able to make an engineered beta adrenergic receptor, which could be activated by a synthetic molecule, in this case a Merck compound, L185870. Turns out that this compound is inactive at native beta adrenergic receptors, but is able to activate a mutant receptor where this aspartic acid 113 is replaced by serine. And since then, a number of others of these chemogenetic technologies have been developed. I just highlight two sort of very important papers. First was the design of what were called allele-specific inhibitors to modulate protein kinase signaling. This was invented by Kayvon Shokat at UCSF. And then specifically designed G protein coupled receptors, a technology that was sort of perfected by Bruce Conklin. But I just want to point out that the first engineered GPCR was developed by Kathy Strader at Merck. And so Bruce Conklin's technology he called RASL. So these are receptors activated solely by synthetic ligands. And they've been useful in a number of, historically, in a number of models. You can see here from an article Bruce and I wrote in Nature Methods some years ago that RASLs have been used to, for instance, by Charles Zucker's lab, deconstruct the receptors responsible for sweet and umami taste when they were expressed either in sweet taste buds or bitter taste buds in some really important papers that were published in 2003 and 2004. And these papers are historically important because they were the first use of synthetic biology technology to successfully deconstruct behaviors. This was actually some years before optogenetics technology. Now, before then, Bruce's lab had shown that the RASLs, in addition to having this utility for the central nervous system, also could be expressed in other tissues. Now, the main sort of shortcoming of this technology was that just the overexpression of these RASLs frequently caused a phenotype in the mouse. And the other thing was that the ligand that was used was not chemically inert. So it was a nice proof of concept, and I think these studies by Charles Zucker's lab really are important as the first sort of to change the paradigm of how we deconstruct the neural code. But the technology was not particularly broadly useful. So my lab had been, as I said, had been interested in this really for many years, going back to the early 90s. And we had tried, mainly unsuccessfully on multiple occasions, to make engineered G-protein coupled receptors that might be useful, but had failed. And so a very talented postdoc came to my lab, Blaine Armbruster, and we thought that rather than starting with the receptor, perhaps what we needed to do was to start with a drug which was inert, and then try to engineer the receptor so it could be activated by the inert drug. And we started out with muscarinic receptors because we knew a lot about their pharmacology and the structure and function of them, and also because, as I'll show you in a minute, they can be expressed in yeast. And so the idea was perhaps we could make an engineered muscarinic receptor which could be potently activated by this drug here, clozapine N oxide, which is pharmacologically inert, but not by the endogenous ligand acetylcholine. And the question arises, why did we choose clozapine N oxide? Well, the reason is that it has very good drug-like properties. So we were imagining that if we could achieve our goals, then we ultimately would like to take it to humans. And it turns out that clozapine N oxide has excellent oral bioavailability, so ultimately it can be given as a pill. It gets into the brain very well, and it's safe in humans. So we realized that if we were able to make a receptor that could be activated by clozapine N oxide, ultimately we could translate this technology to humans. Now, the other thing about clozapine N oxide is it has very rapid pharmacokinetics, so you can see that here. This is from a study that was published by my lab and Jürgen Wess's lab a couple of years ago. And there's been some concern with this technology that clozapine N oxide might be converted to clozapine. And I think this probably does occur in humans to some extent, but does not occur in laboratory mice to any extent at all. So there's basically no interconversion. So it's very useful in that respect. Now, the other thing that's quite interesting about clozapine N oxide is that it's pharmacologically inert. So this is another profile that we did of clozapine N oxide there, shown in the red, versus clozapine at a large number of human brain molecular targets. And you can see that although clozapine has this very complicated pharmacology interacting with many receptors, CNO is completely inert. So we realized that if we could make a receptor activated by CNO, it would likely be very, very useful. And the question was how to do this. So we made a number of attempts to do directed mutagenesis, and that didn't work. But we realized that potentially we could use the power of yeast genetics to evolve a receptor to be activated by CNO. Now, it turns out in yeast there's a single G-protein coupled receptor. There's a lot known about this pathway, but just to cut to the chase, it turns out that you can eliminate the yeast G-protein coupled receptor and put in virtually any human G-protein coupled receptor with a engineered G-protein. And then when the receptor is activated, you can induce the expression of a protein called His3, which allows the yeast to grow on media lacking histidine. So basically, when the receptor is activated, the yeast grows. So we realized that we then had a very powerful selection procedure. So the idea was, as shown in the next slide here, if we did random mutagenesis of the muscarinic receptors, and we can make hundreds of thousands or millions of mutant receptors by error-prone PCR, and then simply put the mutant muscarinic receptors into yeast. And then the key was just to grow the yeast on media that contains clozapine oxide. Any yeast that were expressing a receptor, which is activated by CNO, then grow. So you have this very powerful selection procedure, sort of life and death for the yeast. And then this process can be repeated many times until you have the receptor with the appropriate pharmacologic properties. And what we were looking for was a receptor that was activated by very low concentrations of CNO, in this case nanomolar concentrations, and completely insensitive to acetylcholine. And so this technology has been described, as you can see. We have a very nice protocol that was published in Nature Protocols about 4 years ago. Now, it turns out that, as luck would have it, there were 2 point mutations that we made initially in the M3 muscarinic receptor, which allowed it to be activated by CNO. And it turns out that these 2 residues that were mutated are conserved among all muscarinic receptors throughout phylogeny, going from Drosophila all the way up to humans. This allowed us then to make a family of muscarinic receptors, all of which are activated potently by clozapine oxide, but completely inert. Now, the other thing that, if I just go back here, the other thing that this selection procedure allowed us to do was to screen out mutants which had basal activity, because we knew from the studies of the RASLs that if the receptors had high levels of basal activity, they likely wouldn't be useful. And these could easily be screened out during the initial screening that we did. So we had these, and just to summarize a large amount of published data, this has led to basically 3 types of engineered receptors that we have dubbed DREADs. DREADs for designer receptor exclusively activated by designer drug. And we distinguish these from RASLs because of their unique properties. So we have one which we call HM3DQ, which increases intracellular calcium, and as I'll show you, induces firing in neurons. One called HM4DI, which mediates neuronal inhibition. Another that activates G-alpha S, increases cyclic AMP, and can modulate signaling. We have a website which describes all these, dread.org. All the plasmids that I'm going to describe, all the vectors, are available through AdGene, and viruses that express them can be obtained for a nominal fee from the UNC Vector Core. So they're widely available. Now, since we developed this technology, literally hundreds of labs around the world have adopted it. I would say second only to the adoption of channel rhodopsin. And this just summarizes some of the more interesting papers that have been published. So there have been a number of papers showing that DREADs can be used to regulate neurons that encode hunger, can be used to deconstruct processes related to memory, altered striatal neuronal plasticity, regulating serotonin neuron firing, regulating neurons responsible for pain, and also regulating circadian clock rhythms and other things. And there, as I said, there are many, many papers published on this, and there will be 2 review articles in annual reviews of neurosciences and annual reviews of pharmacology summarizing this information. Now, what are the practical considerations of DREAD technology since there are so many labs using it? Well, unlike the opsins, where you need very high levels of expression to gate neuronal firing, high levels of DREAD expression are absolutely not needed. And the reason for this is that the efficacy of DREADs is limited by intrinsic G protein coupling efficiency. And it's really the downstream components that limit efficacy of the system. And maximum activation and silencing can be achieved really with very low levels of expression. The other thing to realize is that it's essentially impossible to overstimulate the system beyond that achieved by endogenous GPCRs. So occasionally people will be concerned that maybe you're overactivating the system artificially, and really this is impossible to achieve. Now, the consequences of this is that when you have very high levels of receptor expressed, because of the principle of receptor reserve, really adding more ligand is essentially useless. Enhancing DREAD expression really augments the response only until the system is saturated, and the only thing it really does in terms of ligand efficacy is enhance the potency, and that's shown here on the right with increasing levels of DREAD expression. You basically shift the dose response curve to the left until you achieve basically a saturating effect. The other practical consideration is that when DREADs are overexpressed, it diminishes desensitization of the system. Now, what I'd like to do for the remaining time is just go through some uses of DREAD technology, starting initially with the G-alpha-Q DREAD. Now, when we first made this, it was not entirely clear to us what the utility of increasing intracellular calcium was, but as I'll show you, what it appears to do is increase neuronal excitability and in many neurons induce burst firing. And one of the first studies that we did was to basically create engineered mice which express the DREADs under control of an inducible promoter. In this case, we used a tetracycline inducible system, and the idea is that when the DREAD is expressed under control of a tissue-specific promoter, we get expression of the DREAD in a particular tissue, and then we can give the mouse clozapine and oxide here and induce some sort of physiological response. And we wanted to use a promoter that allowed us to turn the DREAD on or turn the DREAD off, because it wasn't entirely clear at the time whether overexpressing the DREAD would have any sort of baseline consequences. It turns out it doesn't. But this illustrates how, using a CAM kinase 2 promoter, we got very, very high expression in pyramidal neurons in the cortex and hippocampus, and this could be turned off by doxycycline, and it works quite nice. And for anybody that's interested, those mice are available in Jackson Labs, and Mark Mayford published a very nice paper in Science a couple years ago using these mice, so they work quite well. The other thing you can see is that the DREAD is very highly expressed throughout the cell. You can see here the apical dendrites and cell body, and you can also see axonal processes as well. So it's targeted very well. Now, the question was, what happens when the DREAD is activated? And we thought it might induce depolarization. And to investigate this, patch clamp studies were done in hippocampal slices from mice that were expressing the DREAD or mice that were not expressing the DREAD. And these were done by Georgia Alexander, who at the time was in Jim McNamara's lab at Duke University. And you can see here on the lower left here that when CNO was applied, we got this very brisk depolarization. Notice how quickly it occurs after CNO administration, really within a few seconds. But in slices that were not expressing the dread, CNO had absolutely no effect. But the effect could be mimicked by activating endogenous muscarinic receptors with carbachol. Okay? So a very similar type of depolarization. Now, the other thing that was noticed by Georgia was that, so in those previous studies, the electrophysiological experiments were done in the presence of tetrodotoxin, which blocks neurotransmitter release. And what she found was that when tetrodotoxin was removed from the bath, not only was there this depolarization, which occurs within a few seconds, but also neuronal excitability was enhanced, and in this case, there was bursting, this very nice bursting of hippocampal pyramidal neurons. And this is basically a physiological type of asynchronous discharging of neurons, which is quite distinct from sort of the synchronized bursting of neurons that can be induced by optogenetic technology, and basically utilizes pathways that are present in essentially every neuron. Now, the other dread that we made is coupled to G-alpha I, and we postulated that since G-alpha I coupled receptors ultimately can activate G-protein inwardly rectifying potassium channels via beta-gamma mediated activation, that these might induce neuronal silencing. And in fact, in the original paper, studies done by Stefan Hurlitz's lab showed that bath application of CNO to hippocampal neurons in culture induced this very brisk hyperpolarization. You can see there within 20 to 40 milliseconds or so after bath application of CNO, and then and silencing of the neurons. Then when the CNO is removed, basically the neurons come back. So it turns out that the M4 dread also nicely induces hyperpolarization and some silencing of neurons. There also was data presented at the neuroscience meeting in last November by the Genelia Farms group, that the HM4 dread may also induce synaptic silencing by inhibition of neurotransmitter release. So it may actually have a dual action of basically causing neuronal silencing by hyperpolarization and also synaptic silencing. So with these two in hand, what I want to do now is take you through a little case study of how these have been used to deconstruct feeding behavior. And for many years, it's been known, of course, that various neurons in the hypothalamus, primarily in the arcuate nucleus, paraventricular nucleus, lateral hypothalamus, and so on, can regulate feeding behavior. But it's the field has been quite controversial up until the past few years, and it was not entirely clear which particular neurons are responsible for regulating which particular aspects of feeding versus metabolism, and so on. And so in a collaboration that was done with Bradford Lowell's group and headed by Mike Crashes, who was in his lab at the time, let me just say that he's currently has his own lab at the NIH. We took the following approach. What we did is we decided that we could probably activate or inhibit these neurons using DREAD technology. And we took advantage of these double inverted repeat viral vectors that were initially invented by Scott Sternsen's lab. You may know them as DIO vectors. And they have the interesting property that you can use them for a particular cargo, and in the absence of cells expressing Cre recombinase, the cargo is expressed in the antisense direction, so it's basically not expressed in the neurons of interest. However, if you inject the viruses expressing this cargo in neurons, for instance, AGRP neurons, agouti-related peptide neurons, that are expressing free recombination, you get recombination and then expression of the dread. And in fact, this works amazingly well, really for dreads as well as for opsins and many, many other cargos, and is sort of now ubiquitously used for expressing cargos in individual neuronal subtypes. And so what Mike did then was to take the M3 dread, you can see here, M3 dread virus, inject it into mice that have agouti-related Cree, and he got very nice expression here exclusively in AGRP neurons. So you can see that in the immunofluorescence of M-cherry fusion proteins very well in AGRP neurons, and actually no expression really anywhere else in the brain or in any other cells. So it's very, very selective. And when slices were made from the arcuate nucleus area, and those neurons were individually recorded with microelectrodes, when CNO was bath applied, we got this very nice depolarization and great increase in neuronal excitability. And notice that this is an asynchronous discharge. This is sort of the neurons firing the way they normally would if they were activated by metabotropic signaling. And the consequences of this were quite astounding. And you can see here in the red line that when the AGRP neurons were activated, the mice basically ate in a very voracious fashion. And within about 2 hours after CNO administration, they had ingested about 1.2 grams of food. And this is quite amazing, because the mice only weigh about 20 grams, so they're ingesting a considerable amount of their body weight in food in this very short period of time. The other lines basically just show the various controls. Now these findings were remarkable, basically, for the extent and duration of food intake, and they are very reminiscent of studies that were performed simultaneously by Scott Sternsen's lab, where he had done essentially the same experiments, but activating neurons with channel rhodopsin, and the degree of stimulated eating was essentially the same in both papers. So it was nice to see that. Now, in addition to increasing the appetite of the mice tremendously, it also increased basically the motivation of the mice to eat, and that's shown here using what's called breakpoint analysis, where mice are trained to basically do a nose poke to get food. And you can see in the lower left-hand corner that mice that were expressing the dread and given CNO have this huge increase in the breakpoint, which indicates they're tremendously motivated to eat. And the increase is essentially the same as seen in mice that are being starved. So this tremendous increase in feeding. As well, there was an increase in locomotor behavior. We think this is because the mice were searching for food. Now, since then, a number of studies from a number of labs, and I'm just sort of showing the references here, have all recapitulated this finding that activation of neurons with HM3 EQ induces neuronal activity. This was seen in cortical neurons, hippocampal neurons, striatal neurons, hypothalamic neurons, and so on. You can see many of the papers there, and obviously many, many more, but these are sort of the most high-profile papers. So the technology works quite well. The next dread I'd like to describe here briefly is the HM4 dread, which induces neuronal inhibition, probably by 2 ways. One is by hyperpolarization, and the other is by inhibition of neurotransmitter release. And as I mentioned in our very first studies, we were able to show that activation of it silenced pyramidal neurons really quite briskly and within a few tens of milliseconds after CNO administration. So the effect is quite robust. And again, this is mediated by beta-gamma mediated activation of GERK channels. So we wondered, so I wondered, since the effect of the M4 dread is rather indirect, whether this would be something that would be seen in other neurons throughout the brain. And what I'm going to do is just summarize many, many studies that have occurred since then, showing that this appears to be general. So a nice paper showing that silencing parvalbumin neurons in the cortex occurs in vivo by Kuhlmann et al. Another paper by Rui Costa's lab showing that cortical neurons can be silenced by HM4 dread. A number of papers now showing that direct and indirect pathway medium spiny neurons in the striatum can be silenced. Various, now many pathways or many papers have now shown that various hypothalamic neurons can be silenced by HM4 dread. And really, this is I would say, a very, very active area of research. Now, papers coming out every couple of weeks now showing effects of dreads on hypothalamic neuronal activity and feeding behavior and other sorts of phenotypes. There also is data showing that neurons with long afferents, for example, thalamocortical afferents, can be silenced by HM4 dread. Cholinergic interneurons can be silenced by HM4 dread. And serotonergic neurons. So this is a very nice study by Ray et al. In Science showing that serotonergic neurons can be effectively silenced by HM4 dread in vivo. So really a number of studies have demonstrated now the utility of the M3 dread for activating neurons, the M4 dread for inhibiting neurons, and as I'll show you now, the GS dread for modulating signaling. So we'll go to that one next. So there have now been several papers showing that the GS dread can modulate denylate cyclase activity in vivo in neurons and elsewhere. And I just cite just 3 of the papers that have occurred. Nice paper by my lab here, as well as other labs. And it's likely that many, many other studies of these sorts will be coming out in the next year. And in fact, there's a paper describing the utility of dreads that appears in the literature every day or so now. So this is a widely utilized technology. Now, in addition to the brain, central nervous system, and neurons, there have been a number of reports now showing that dreads can be used in the peripheral nervous system. Really nice paper by the Anderson lab, Deconstructing touch. And these are mass-related G protein coupled receptor expressing neurons in the periphery and GRPRX. Nice series of papers coming out from Jürgen Wess's lab showing that dreads expressed in pancreatic beta cells can regulate insulin secretion, and dreads expressed in the liver can modulate various metabolic responses. Nice series of papers from the NIH group showing that dreads, when expressed in triple-negative breast cancer cells, can modulate the metastatic phenotype. And now several papers showing that dreads can be expressed specifically in astrocytes. Generally, it's the GQ dread, and that this, in induces astrocytic activation and of course has multiple effects on neuronal activity. Intriguingly now, DREADs have been able to be expressed in Drosophila, so there are several fly lines expressing DREADs that work quite well. My lab has created a number of mice that express DREADs either constitutively or inducible. They're all available through Jackson Labs. And as I mentioned, all of the vectors are now available through Agene, and viruses are available through the UNC Vector Core. So to conclude, the DREADs allow for a very effective remote control of cell type specific signaling. This can result either in excitatory and inhibitory effects on neuronal firing. This has been used and continues to be used to deconstruct neural correlates of behavior and is equally effective in non-neuronal cells. The system has now been validated through multiple inducible tissue-specific transgenic approaches, back transgenic approaches, straight transgenic approaches, multiple virally mediated gene transfer approaches, HSV, lentivirus, AAV, and so on. There are a number of resources that my lab has created for these. We have a site, dread.org, which has protocols, plasmid maps, available vectors, and constructs. I've recently started a blog which allows for late-breaking information for DREAD users, which is shown there. And then the NIMH chemical synthesis program is able to supply clozapine and oxide free to qualified investigators, although it's also available commercially. And so that's a nice resource. And a number of AAV-based viral technologies are now available through UNC Vector Core. And so I'd just like to thank the funding agencies. So all of the work in my lab is funded by the NIH, primarily by the National Institute of Mental Health. The work on salvinorin A and opiate receptors is funded by the National Institute of Drug Abuse. The NIMH Psychoactive Drug Screening Program funds all the screening and profiling. And let me just say that this is a free resource available to qualified investigators. So anyone out there who has interesting compounds, we are happy to screen them. It's free, confidential, and IP neutral. And then I mentioned collaborations with Ray Stevens' lab, primarily Daniel Wacker, Chan Wang, and Huishan Wu. Daniel's now a postdoc in my lab. And of course, Mike Crashes, who provided some of the first in vivo validation of DREADs when he was in Brad Lowell's lab at Harvard, and Mike now has his own lab at the NIH. And this is just the DREAD wiki page. And finally, just to let you know that if you'd like to obtain continuing education credits, here's where you can go. So that's all I have to say today. Thank you so much for your attention. And we have probably 4 or 5 minutes here for questions if there are any. And I'll wait 2 minutes. If there are no questions, then I'll hang up here. All right, I think that's it. Thank you very much. Bye.